So thank you for the opportunity. This came about some serendipitously. We were having a conference call about something completely separate, and then Michael wrote me into this, and I see that I'm not the only one.
I think that's pretty much how every one of us got in.
Yeah, I'm starting
I
to see
have all your
a theme.
emails and cell phone numbers.
And we just locked the doors.
That's
And then to make things even more interesting, he asked me to actually talk about two different things. I wear two hats. My full-time job is actually at the University at Buffalo, where I try to craft more partnerships with industry with the idea of advancing the knowledge that we're developing at the University at Buffalo. And on the side, or maybe my other full-time job, is to try to develop this treatment for an extremely rare indication. I'll tell you a little bit about that too. So I figured that it is a little bit hard in just 20 minutes to really tell you the different verticals that we have and how they plug in. So our motto really is win-win-win, and here the three wins are represented by the state of New York. They are our main supporter as far as funding goes. The University at Buffalo, obviously, that there's a win in there for us, and then the life science ecosystem that we're trying to nurture and develop in upstate New York. And it is still certainly emerging, but perhaps if there's anything that we can share with you that would be helpful towards your biotech project here, I'm an open book, so ask away. So our activity really relates to making sure that we externalize as much of whatever it is that we're working at the university to our region or the state, and of course we'll be opportunistic, and if we can do a deal with Takeda, we'll certainly do that. So the idea is really to provide faculty and students the opportunity to engage with companies, again, regionally or at least in the United States, and to really learn what the real world is about, rather than just being exclusively located at the university going to class. We try to support the commercialization of intellectual property. We support startup companies that are actually either in our region, working with us with, for example, our genomics course, proteomics course, the supercomputer, etc., and try to provide them with some additional support. And that can be very, very different. It could be finding them a CEO, it could be finding them an entrepreneur in residence, supporting their application for SBIRs, STTRs, etc. And ultimately, we want to help the companies that are there stay there and grow and be successful. And there, of course, New York State is very interested in being able to capture additional dollars through taxes. So that's their main interest there. And then I'm gonna go pretty fast through these slides, I guess in the interest of time. I want to make sure we have time for questions at the end. And this is one of our customers. We have been working with Irfan, their CEO of Circuit Clinical, for a number of years now. And this is a company that is specializing in being able to bring a clinical trial to a small provider of care anywhere in the Western New York region, and he's expanding to other geographies right now. The idea being that a provider of care does not have the time to figure out how to run a clinical trial, does not want to follow the patients, does not know the protocols, does not want to engage with the companies, etc. So Circuit Clinical brings a team to their side, goes through their medical records, and figures out, you have 15 patients for this clinical trial with Roche. If you're interested, we'll set up all the overhead, the back office, etc. And all you need to do is to give us an extra 15 minutes with that patient at your office, and we'll do what we need to do for that clinical trial. So again, it's trying to expand the breadth of clinical trials. And how we have worked with Irfan. So again, this is a number of the different verticals, and just in the interest of time, I'm going to fly through them. But if you're interested in learning more, my email address is somewhere in here, and I'm sticking around for a while. So feel free to ask questions. But at a high level, we have a Center for Computational Research. This essentially gives Circuit Clinical all the computational horsepower that they might need to go through all those medical records, identify patients, do the cross-matching between the company, clinical trial interest, and the patient populations that we have in Western New York. They have partnered with an economic development incentive that I am supervising, called the Buffalo Institute for Genomics and Data Analytics, in which it was a $47.5 million investment from New York State. I still have $20 million to commit to companies growing. It is limited to Erie County, so if any of you have companies interested in perhaps locating back to Buffalo, it's a beautiful place, and it is not always snowing, I promise. I am from Madrid. I did not move there for the weather. So. But nevertheless, it's a great place to be at, and we essentially provided them with access to specialist equipment and to the Center for Computational Research at no expense to them. The idea being that as long as they hit those job goals, I will forgive all the money that we have deployed in their company. They're also a startup New York company. This is a tax incentive program from New York State that essentially enables Circuit Clinical to have their employees not pay New York State income tax for a period of 10 years. We have helped them find a home close to us. This is particularly helpful because they're always working with the Center for Computational Research. So if they have a number of jobs and something seems to be going wrong, they can very quickly go to a computational scientist and figure out what's happening. They have received a CAT award, Center for Advanced Technology. This essentially is matching funds. So we'll put in $25,000 of our own money, they'll put in $25,000 of their money, and we'll find an investigator to tackle a specific problem that they are encountering. And they are participating in the Career Experience Program, where we provide interns that are in high school, actually with the opportunity to go to a company to get a feel for what it would be to work in a STEM field. And this again has to do with trying to get more students interested in the university and going into STEM fields. Athenix is one of the companies that has made the most noise in our ecosystem. They had an IPO, I believe, a year or 2 years ago. They're currently 1.2, 1.3 billion market cap, so they're, for our region, they're the real deal. They started with a small discovery from this individual who actually taught me chemistry 1.5 million years ago. And they are now setting up a manufacturing site somewhere in upstate New York, and also they have several locations in China, which is where many of their investors come from. Similar to Circuit Clinical, they have partnered with the university in a number of ways. They actually started doing a lot of their chemistry in our incubator space. They took on a license for the intellectual property that was developed at the university. They have participated in a number of different company events at the Center of Excellence in Bioinformatics and Life Sciences, which is where we are located. They have also received a number of CAT awards. So these again are those matching funds. I believe they have received maybe like $100,000 to $200,000 worth of this type of funding, and this has really helped them advance again their company until they became a public company. So that's a great success story. Another similar story is Cytocybernetics. These entities developing a medical device that I'm gonna fly through again. They are a spin-out, so again, the originating intellectual property emerged out of the university. They took a license to it. They went through, or they benefited from an initial award from the SUNY system. Then they obtained another matching fund from our institution. They went through a pseudo I-Core program at the time, which essentially enables the company with how to find customers, validate their idea, etc., before really becoming overly interested in it. And they participate in a different economic development incentive opportunity that we have called WIN, or the Western New York Incubator Network, in which we can actually pay for consultants to actually work with these companies to answer specific questions. Many times it's a regulatory person trying to figure out, is it a PMA, 510K, etc., which was the case in this example. They also are a participant in Startup New York, so they save 10% more or less of their taxes that would otherwise go to New York State. And they frequently participate in events that we organize in the region to showcase who is doing what, spread the word, and make sure that they are in front of the investors that we have in Buffalo. So I know I went super fast through this. There's a lot of different economic development incentives. Generally speaking, if you're working in the life sciences and big data, we can work with you no matter where you are, as long as you're in New York State. We have probably 1 or 2 different economic development incentives that we could put to work. If you're willing to locate, relocate, or expand in Erie County, then I have more real money to put to work potentially with any of you. And there's a few conversations that, just based on today, I'd love to have. So I'll be following up with some of you. And here's my email address and contact information. Again, any questions on how things start to mature in Buffalo, if we can save you some time, we'd love to do that. So just let us know how we can help. Any questions on that side?
Are you going to get a little bit more into the bioinformatics side and what that means, what that Center of Excellence
Sure,
is?
I can. So the Center of Excellence in Bioinformatics and Life Sciences houses a genomics core, a proteomics core, an Institute for Healthcare Informatics, a biorepository, and a supercomputer. These are the 5 assets that we used about $15 million from New York State to establish and to get going. And we provide those resources internally, obviously, to our faculty, but also to companies that want to work with us. So if you are a New York State company and you are in need of some computational capacity or sequencing some samples, etc., we are able to support that type of activity. The CBLS, which is the Center of Excellence, also has incubator space in the second and third floors. So companies are actually co-locating with us, and there's wet lab space as well in there. The idea being to continue to promote this type of collaboration. So generally speaking, as far as the computation, I'm afraid I'm fairly ignorant on the capacity. I know that we're spending an additional $1.1 million in GPUs when we are turning the supercomputer into more of a machine learning and AI type core facility, I guess. So those resources are for your benefit here. If you're a New York State company, again, we'd love to find a way to work with you and help you develop products, solutions, etc. All right, so now I'm going to change topics drastically. This is what I do for my other full-time job, or in excess of full-time at this point. It is a devastating disease. We have heard some very complicated disorders, and this perhaps is a similar one. It is an extremely rare disease, and we'll get into that in a few slides. I became involved with it after doing some consulting on the side for a European university, and they were working on this gene therapy for the disease called mitochondrial neurogastrointestinal encephalomyopathy, or MENGI. And I don't know if perhaps you guys have seen any patients with MENGI before. I'd be curious to hear. Probably not. Yeah, not surprised. So as far as getting very quickly into the mechanism of action, in this disease, these individuals are missing one critical enzyme involved in the DNTP cascade or pathway, and that's called thymidine phosphorylase, which actually gets rid of this thymidine here, which is going to become a toxic metabolite in patients that do not have this enzyme. So that's what we see here. We unfortunately have an increased presence of DTTP, and this increased excess of DTTP leads to the depletion of DCTP. Mitochondria are not as savvy as our nucleus in a way, and they cannot fix their own mistakes, so they continue to incorporate the wrong nucleoside into their genomes, and over time, these mitochondria become defective, they lose function, and ultimately the patient most often dies. There are 86 mutations that describe this disease, so some of these mutations may be more severe, while others are fairly lethal. This is what a patient with MENGI looks like. This individual has been healthy, most likely, up until age 16 or so, but he was always the skinny kid that could not gain weight. Perhaps there was a little bit of vomiting every so often for unknown reasons. He may have been diagnosed with a number of diseases that were obviously inaccurate, and unfortunately, as time goes on, this individual is going to need parenteral nutrition. The gastrointestinal function is going to be very limited until it arrests fully, causing dysmotility issues, and unfortunately he's gonna die from the disease probably in another 20 years or so. So it's a very slowly progressive disease. These kids are very fragile, and the GI complications are generally what causes the death. Neurological manifestations do happen first, so the first thing that you might see is that the eyelid cannot stay up anymore, and then as the disease progresses, wherever you see more muscle activity, there tends to be more problems. This is the disease progression looking at a total of 102 patients. So as you see, up until age 16, 17, 18, the symptoms have been somewhat minor or at least manageable. But once you get to the third decade, that's when you see the drastic reduction in survival. So most patients will die by age 40 or so. And again, this is due to gastrointestinal complications. This is the data that got me the most excited and why I decided to figure out how to make it work. So when you look at parents of individuals with MENGI, these individuals actually have 25% activity instead of having 100% activity. And the reason for that is that thymidine phosphorylase forms homodimers. So in the case of wild-type individuals, most likely like all of us in this room, we all have two good copies of the allele, so 100% of the time we have functioning thymidine phosphorylase. In the case of parents of MENGI disease, they have one good allele and one bad allele, and that actually leads to 4 possible combinations, 3 of which are not going to be functional. So from my perspective, knowing that the parents are healthy and they live long lives without any symptoms, and that patients do not have any activity of this enzyme, thymidine phosphorylase, the window of opportunity is here. Effectively, if I can deliver enough activity by enzyme replacement or any number of different approaches, and I can get to this level, presumably the disease will be stalled. Now, the mitochondria that have incorporated mutations will continue to be there, but at least the disease will be arrested And we know, because some of these patients have received bone marrow transplants, and although very few of them have survived, again, if you imagine such an individual receiving an allogeneic stem cell transplant, it's a devastating thing. So, in fact, all patients over the age of 30 have died from the bone marrow transplant, and the HLA match, if it was less than perfect, 10 out of 10, they also succumb to the transplant. Nevertheless, if we can deliver enough activity, somehow, to get to this level, it should be at least curative or at least put a break on the disease. So that's why we thought that it was a great gene therapy target. And here we have, all right, this slide got a little bit screwed. Okay, doesn't matter. So in a way, some of the early clinical data, and if it's of interest to you, I have a big slide deck that I can share with you. We were looking at a double knockout mice that essentially did not have two proteins in mice, and that is because there's a little bit of a difference between how mice do things and how we do things. But effectively, we delivered an adeno-associated virus to them that was expressing thymidine phosphorylase. This was an AAV virus that would target specifically the liver. It's very similar to a different virus that a company is developing, and it's been quite successful so far in hemophilia. The idea being that if this virus was able to express sufficient enzyme in the liver, biochemically, we should see thymidine and deoxyuridine be reduced. And behind this text here, theoretically, you were going to be seeing some data that you are not going to see today. In any case, we have done a lot more work on this disease. There's been many years of work by the scientists leading this effort. We know that the expression of thymidine phosphorylase is limited to the liver. We do see, however, that the reduction of those metabolites is systemic. So, for example, if we look at the brain, we know that there is no increased concentration of those toxic metabolites in the brain. And we know when we follow these mice for 2 years, unfortunately, mice live about 2 years, and it is not long enough for us to see the degenerative disorder because it takes so long in humans. But nevertheless, we see that it is a dose-dependent efficacy. We know about how much virus we need to deliver to make sure that the efficacy is there and it does not get diluted. And when we do necropsy-type studies, we do not see any surprises looking at the different tissues. So far, that's quite encouraging. We have been doing some work this past year in trying to optimize the vector to make sure that we have the Ferrari going into the clinic instead of a crappy version. And we now have looked at complementary conformations versus not. We have looked at a number of different promoters, and we can also optimize the sequence of the thymidine phosphorylase to ensure that every single viral genome that gets into the cell expresses as much protein as possible. And we have, I'll fly through this. We essentially, if we look at these red boxes, and we look at the blue versus the purple was the vector that we were very excited about until we developed the blue one with the new promoter. And you can see that the blue one is better able to control the disease and bringing the levels of this toxic metabolite back to wild type quickly, only 1 week after the treatment. And when we look at 4 weeks, now we see that the purple one, which is the one that we were developing, is efficacious, which is good, is what we expected. But the best part of it is that the purple one, and most of the data that we generated with the version 1.0, was using 5 times 10 to the 11 or higher doses. And what we're seeing with the blue promoter is that, in fact, this promoter seems to be at least 10 times more efficacious. So the reasoning behind this is that now, if we can get the same amount of virus into the liver of these patients, we'll actually have 10 times more activity. So that is quite encouraging, and we're looking forward to getting it into the clinic. And here's the challenge. So we've been talking about rare diseases, and I'm afraid this is perhaps the rarest of them all, other than perhaps AJ. So it is a disease that when we look at the experience in a number of different countries, this is the number of patients that I am actively following right now in those countries. I know that perhaps the best number is this one for Italy, because there was an Italian research group that actually sent out surveys to geneticists, to neurologists and gastrointestinal doctors, trying to figure out, hey, do you have a patient that looks like this? And they will have like some symptoms, some pictures, etc. And they were able to identify 7 of them. So if I had to put my hand on the fire, I would say that the prevalence most likely is going to be 1 for every 8.6 million people. So nevertheless, that's still fairly small. Looking at incidence, which is quite different from prevalence, as many of these individuals may not make it, and they may never be diagnosed. Our best estimate is to have 1 to 2 patients for every European country, and based on those estimates, we're probably somewhere between 7 and 15 in the United States. So a total, we're looking between the United States and Europe of 20 to 40 patients, more or less. This, of course, presents a unique number of challenges for fundraising because not every investor is interested in such a small indication. Nevertheless, this is a version of the deck that I'm using to fundraise with different versions of investors. Most of what we're missing at this point, we have the clinical candidate, we have the contract manufacturing already lined up. We know the company that is going to be doing it, at least so far. And we know that process is going to cost us probably another $2.5 million or so. We know the toxicology type of experiments that we're going to need to be doing to make sure that we can get to the IND. And that's going to run us another $500,000 or so. And ultimately, the clinical trial, we have theoretically 2 sites. I am confirming that I have enough patients only in France and in the United States to do a multinational clinical trial so that we can get to the market both in the United States and in Europe within hopefully 5 years for approximately $12 to $15 million. Some of the things that I am always looking, and of course, for you in the audience that may have friends or colleagues interested in this, I'm always looking for patients, providers of these potential patients, and there's a particular gap that I have, which is trying to find some support in regulatory consulting. So I know there's at least one conversation that I want to have with some of the people here. Lastly, this is the summary of the opportunity. We know that these toxic metabolites, although they happen systemically, they actually diffuse through membranes very easily. They're water soluble. So if we put enough enzyme in the liver, it's going to act as a sink and therefore clear all the toxic metabolites systemically. We know that heterozygotes, or the parents of these patients, have 1/4 of the activity. So that is encouraging in that if we deliver 1/4, we presumably would arrest the disease. Perhaps we need to deliver less, but at this point, 25% seems like a good goal to get to. We have a precedent, very similar virus in hemophilia being developed at Spark Therapeutics. Again, it's an AAV28, so I'm very closely following them and talking to them about some of the nuances of their development path. It is very likely going to be a single-phase clinical trial, which will have a maximum of 18 patients alone, which is why the clinical trial is relatively inexpensive and fast, based on how rare this disease is. And there has been a protocol assistance meeting with the European Medicines Agency back in 2015 that was favorable, so we have a clinical trial structure, etc., that has been agreed to. Regarding the proprietary positioning, we have orphan designations in both Europe and the United States for version 1.0. We are submitting the same for the clinical candidate that we are excited about. And we do not have any patent protection, and I am very proud to say that, and I have no problem with it, because it's such a rare disease that just doing patent prosecution in all those different countries would actually be quite expensive and painful. And it is a very small indication. Assuming that we can get to the market for 7 and 12 years of market exclusivity, I seriously doubt that a real company might actually try to have a superiority trial when comparing this product. And obviously, our biggest challenge is the incidence. It is beyond ultra-orphan, as you can see. So there's a number of challenges on how do we come up with the money to in fact, deliver this. So I put this slide in the background with many doors. I am a big believer in knocking on absolutely every door I can. So if you think that you have a friend or someone that might be interested in learning more about this, I would love an introduction and walking them through the science. Thank you.